So let's start with the bad news. We're still in the middle of the hurricane, and it's going to go on for a while yet. Nationally, it's a firestorm of COVID activity, as you can see on the map. Hospitalizations are at or near record levels. Over 300,000 people have died in the United States. In Wisconsin, we have seen some reduction compared to our peak in early November, although we are still at a very, very high level of hospitalizations and cases compared to where we were over the summer. So the vaccine isn't going to save us from this in the short term. We have to get through this with the usual measures that we have all heard about. But there is good news on the horizon, especially in the past week. The FDA has authorized one coronavirus vaccine by Pfizer. It is on the verge of authorizing another one by Moderna. I was actually wondering if that would happen before this grand rounds. And so this is the beginning of the end. And there is light at the end of the tunnel, but we still have a ways to go in the tunnel. So one thing that I think is just amazing to reflect on as we think about this is how far we've come in the past 11 months. This started really on January 10th with a tweet from Edward Holmes at the University of Sydney in Australia, who basically shared with the world the sequence of SARS-CoV-2 virus, which had been shared with him by a scientist in Shanghai. And this was the information that sort of then led very rapidly to a sequence of events that over 11 months, culminating on around the last week, physicians and other healthcare workers and long-term residents of long-term care facilities had the opportunity to be vaccinated with a vaccine that had been fully vetted and assessed by the FDA and by independent advisory panels and proven to be safe and effective. So this is really a record in terms of vaccine development. I think the earliest or the fastest vaccine development before this was about 4 years for a measles vaccine. So how does this normally work? I'll just talk a little bit about the vaccine development pathway and sort of what was changed in order to make this happen. Typically, in order to develop a vaccine, a company goes through a long sequential process that often takes a decade or longer to go from vaccine preclinical studies to an actual licensed vaccine that is being given to people. And as you can see in this graphic, first there are animal studies, there are laboratory studies looking at safety and concept, and then it moves into human clinical trials, first phase 1 safety, and then phase 2 for safety and immune response, and then finally phase 3, which is designed to actually show efficacy for preventing the disease. Then it gets submitted to the FDA for review and approval. If the FDA approves it, it is licensed and can be used, and then they go into post-approval surveillance. And throughout the whole process, the FDA monitors this very closely to ensure safety, efficacy, and manufacturing consistency. So what's the difference with COVID-19 and how did it happen so fast? Well, there are a few things to focus on. First, this didn't happen in a vacuum. There was a foundation of knowledge that was built up based on the original SARS epidemic, which happened in 2003. And after that, there was work on some SARS vaccines in animals that were eventually stopped because SARS was eradicated. But we learned a lot about the SARS virus and the immune response to it and what triggers the immune response. So we had that knowledge because SARS-CoV-2 is very similar to the original SARS virus. Same with MERS, which is the Middle Eastern Respiratory Syndrome virus, and there were vaccine work on MERS as well. And in addition, there was work on mRNA vaccines that I'll talk a little bit more about in a couple minutes. The other thing that was done this past year is instead of doing the normal sequential phases of phase 1 to phase 2 to phase 3, they were overlapped. And protocols were written to cover 2 or even 3 phases all at once in order to speed the process up and avoid delays for going from one phase to the next. And another very key component of this was the fact that with support from Operation Warp Speed and the U.S. government, there was scale up of manufacturing even before the human trials were complete. So in other words, people were not put at risk, but money was being put at risk, because if these vaccines were manufactured and then shown not to be safe or effective, all of the money spent manufacturing them would be wasted, of course. And so that's a risk that a company would never take with normal vaccine development. But given the urgency of the global pandemic, that was a risk that governments were willing to take in order to ensure that we had a vaccine for the population as quickly as possible. So I hope to convince you in the course of my presentation that all of this was done while maintaining scientific integrity and did not cut corners in any way. So I want to talk a little bit about FDA regulatory review for COVID vaccines and the difference between licensure and emergency use authorization. Licensure is a very formal process that is governed by statutory and regulatory requirements that are by definition have to take a long time and require complex analysis of preclinical data, animal data, clinical data, and long-term follow-up safety and efficacy data, as well as actual inspection of the manufacturing facilities. Now, typically, even in a fast-track situation, licensure takes at least a year after a company applies for accelerated licensure. But the FDA has the option to issue an emergency use authorization, and it's called an EUA, instead of licensure in the situation where there's a declared emergency situation, which we have now with COVID-19 and have had since March. If the FDA determines that the potential benefits outweigh the risks and there is no available alternative. So that certainly fits with COVID-19. In addition, the FDA has an independent advisory panel called VRBPAC, that stands for Vaccines and Related Biological Products Advisory Committee. These are independent experts. They're not government employees. They conduct transparent and rigorous review of evidence for safety and efficacy. And the VRBPAC group has met. They met on the 10th for the Pfizer vaccine. They met yesterday for the Moderna vaccine. These are open public meetings. The slides and the briefing materials are available on the FDA website, shown at the bottom of this slide. And in fact, many of the slides I'm going to show you coming up are they're actual slides taken from these meetings, because I couldn't do better than what was presented at these meetings. The other thing that I would mention is that the experts on VRBPAC are vetted for conflicts of interest, and at the FDA also the data submitted by the manufacturer is completely re-reviewed and reanalyzed, and so the presentations for the VRBPAC meeting include presentations from the companies. They also include presentations from the FDA, where career scientists at the FDA have completely reanalyzed all the data to ensure that the conclusions presented by the company are accurate and valid. Okay. So I want to cover this slide briefly. These are vaccines in late-stage clinical development in the US and the UK. The first two are Moderna and Pfizer, and I'm going to cover each of those in a lot of detail. I just want to briefly mention three others, because I'm not going to talk any more about those today just due to time. And I think we'll be hearing more about these if they get to the point where they've applied for an EUA. But one is from AstraZeneca and the University of Oxford, and they use a very different technology. It's a chimp non-replicating adenovirus vector. An adenovirus is a virus that causes mild common cold symptoms in humans. This is a chimp version of that virus that would basically deliver the instructions to manufacture the spike protein. And there is a large phase 3 clinical trial underway right now with that. Another one is Novavax, which is a US company. They have a recombinant nanoparticle spike protein with an adjuvant. This is really very similar to a flu vaccine that they have in late-stage clinical development. They performed very well. This vaccine is about to enter a phase 3 in the US and Mexico, and they have a phase 3 underway already in the UK. And then the last one is Janssen, which is part of Johnson & Johnson. and they have another non-replicating adenovirus vector. It's similar to what they have in their licensed Ebola vaccine. This is the only one that's a single-dose vaccine, and that also is in phase 3 studies right now. So you may be hearing more about these other 3 vaccines in the coming months, but I won't be talking more about them today. Okay, so in order to sort of understand the vaccines, it's helpful to understand a little bit about the virus and the immune response. So the SARS-CoV-2 virus has a major surface protein called the spike protein shown in this graphic. And the spike protein is what triggers the immune response and triggers antibodies that are neutralizing to the virus. And it's also what the virus uses to attach it to the ACE2 receptor on the cell membrane. So if you antibodies that bind to this spike protein can inactivate the virus and prevent binding to the cell membrane. And now I want to talk just a little bit about the concept of messenger RNA vaccines, because this is a new platform. It's a new technology in the sense that we have not yet had a licensed vaccine using this technology. Messenger RNA is the genetic blueprint that cells use to synthesize proteins. And in fact, if you have a viral infection, if you have a flu infection, the flu virus gets into your cells and it uses messenger RNA to make copies of the flu virus that then leave the cells and make you sick, and also can transmit the virus to other people. So it's really very similar to that, except in this case, this messenger RNA is synthetic. It's delivered to you through an injection, and it only codes for a single protein. In this case, it codes for the spike protein. So the cells translate this messenger RNA into the protein antigen, which then is expressed on the surface and causes antibody production and a T-cell immune response. The messenger RNA that is injected is just like the rest of the messenger RNA, and it is rapidly degraded after it is translated into a protein, and it is not self-replicating. It doesn't alter human DNA, and it does not get into the nucleus. Both of these products are manufactured by cell-free production, and they don't use any ingredients of human or animal origin. There are no cells, and there's no need to actually grow an actual virus. So that's a real advantage in terms of safety and efficiency of manufacturing. And the cartoon at the bottom, which is from Moderna, just kind of shows how the mRNA is sort of the software that sort of drives the production of the protein, which is the actual output or application. And on the right, you see a schematic of the mRNA, which is inside of a lipid nanoparticle, which allows it to get inside of the immune cells and do what it's supposed to do. So this is a cartoon presented by Moderna at the VRBPAC meeting yesterday. I think really sort of nicely sort of illustrates how this works. So on the left, you see the spike protein encoded messenger RNA that attaches to an antigen-presenting cell, typically in the lymph node nearest to where you were vaccinated. And then inside the cell, that messenger RNA is released from the nanoparticle. It attaches to the ribosome, and then it's translated into the viral spike protein. Again, there is no actual virus. It is just a single protein, and then that protein is expressed on the surface of the cell as a spike protein trimer, and then that then triggers immune response. So as I mentioned earlier, this is not brand new technology, even though we do not have any previously licensed vaccines using mRNA. And with all of the efforts going into this, there's thousands of people and many stories, and I just want to mention one story here. I think it's an important one, and just to give you a sense of sort of the background work of what has gone into this over the past 2 decades that got us to this point. And this is a story of a BioNTech scientist named Catalin Carrico, who was at the University of Pennsylvania back in the 1990s working on mRNA, and she was building on some work from University of Wisconsin scientists who had basically provided some evidence in animals that basically synthetic RNA could be used to get cells to manufacture therapeutics or vaccines. And she was trying to develop that, and but wasn't getting a lot of attention, and no one seemed terribly interested in the work she was doing. And she met up with an immunologist named Drew Weissman, and he was also very interested in this. And they were encountering a number of difficulties in terms of mRNA causing a lot of severe inflammation when it was injected. And in 2005, they discovered an approach to making a slight modification to one component of the RNA called uridine, which is a nucleoside. And they made a modification that's a naturally occurring modification that greatly reduced the inflammatory response and improved the stability of the messenger RNA and the translational capacity. And they published this in 2005, and they published a follow-up in 2008. And it didn't get much attention at the time. And she actually was demoted in her position at the University of Pennsylvania in 2013, and she decided to leave, and she joined BioNTech in Germany and continued to develop the mRNA. And in 2017, they had proof of concept where they actually showed that an mRNA vaccine against Zika virus was protective in animals with a single low-dose vaccination. So over the past 2 decades, there's been work going on in this arena. And in fact, there have been multiple mRNA vaccine trials even before COVID-19. And this is actually from, it's a tweet from Katalin Karikó. And I haven't found this anywhere else, so full disclosure, but she's a pretty reputable source on RNA vaccine trials in humans. And you can see there have been a number of phase 1 and 1 phase 2 study on a variety of pathogens, including flu, rabies, Zika, CMV, and chikungunya. So that's the background in mRNA technology. So now I want to sort of focus on Pfizer vaccine and results from their phase 3 study and the Moderna vaccine. So the Pfizer vaccine was first one out of the gate that received EUA from FDA. The Pfizer vaccine has some unusual characteristics. It has to be stored at ultra cold temperatures, which creates some additional challenges in terms of distribution and handling. It is refrigerator stable for only 5 days. Requires 2 IM doses given at 0 and 21 days. And the phase 3 trials included people aged 16 and up. So now I'm going to show you a series of slides from the VRBPAC presentations on this vaccine. There are a number of studies in preclinical development looking at safety and toxicity in animals that had no concerns. I want to in particular point out the study in the middle of the slide that was a reproductive toxicology study in rats. And they're planning to announce results in December. I have not yet seen those results, although Pfizer has said that they do not expect to have any concerns in terms of reproductive toxicology because this is obviously a consideration for use in women who are pregnant or may become pregnant after receiving this vaccine. Then they have also done challenge studies in non-human primates, and they found that those studies show a high level of protection in monkeys or macaques that are challenged after vaccination. It is clear that the vaccine generates strong neutralizing antibody response both in younger adults and older adults. This is published in the New England Journal of Medicine a couple of months ago, and this is just it shows 2 different vaccine constructs. The one on the right side of the slide is the construct that was actually approved, or excuse me, authorized for use by the FDA. So I'd focus on the right side of this, and you see 3 different doses. The 30 microgram dose is what is authorized for use. And so you can see the neutralizing antibody titers there at 28 days, so that's a week after the second dose, have increased dramatically, and they're generally similar to what we see in human convalescent sera over on the far right in black. And this is in people 65 to 85 years of age, where we are very concerned about immunosenescence and a reduced immune response. So this is very encouraging, and I'm not showing you the data for younger people, but it was very similar to this. So now I want to focus on their phase 2-3 trial demographics and results. They enrolled over 42,000 people in this phase 2-3 trial in a 1-to-1 ratio of vaccine versus placebo. And you can see from this table, there's a good balance by sex, by race, by ethnicity, and by age group. Although I would point out that a relatively small number of people were over age 65 in the trial, which limits the conclusions a bit when we start looking at subgroups. So this is a top-level result on vaccine effectiveness or vaccine efficacy against symptomatic COVID-19 infection among people who were not infected at the time they were randomized and enrolled in the study, or without any evidence of infection up until they received a second dose of vaccine. And that was sort of a pre-specified primary endpoint for the study. So if you look at the top graph, you see on the top row, the overall effectiveness was 95%. And you can see if you look at that by age groups, although the confidence intervals get very wide as we get into older age groups, the estimates of vaccine effectiveness don't go down in any meaningful way, even among people over age 75. But again, small numbers. That's based on only 5 cases in the placebo group. No real difference by sex, no real differences by race, and no real differences by ethnicity or country of origin. And then at the bottom, you see some additional subgroup analyses looking at different combinations of people with high-risk conditions and people without high-risk conditions. And you can see that just in general, people with or without a high-risk condition, at the top, the vaccine effectiveness is basically the same. It's 95%. And then if we look at different age groups and whether or not they have a high-risk condition, again, we see vaccine efficacy ranging from 92 to 100%. Similarly with obesity, we see no difference with or without obesity, and we see no meaningful differences when we look at age group with or without obesity. So all of these really suggest that we're not seeing any dramatic differences based on people who are higher risk or have certain demographic characteristics. This is, I thought, was a striking slide when I saw it. This is the Kaplan-Meier curve that shows the cumulative incidence of COVID-19 in the placebo group shown here and in the vaccine recipients shown here in blue with days after dose 1 here on the x-axis. And you can see that even at as early as 14 days after the first dose of vaccine, we're starting to see a divergence between the placebo group and the vaccine group. The overall vaccine effectiveness was 95% in people with any comorbidity, diabetes, or hypertension, and the overall vaccine, efficacy was 95% in subjects with or without evidence of prior SARS-CoV-2 infection. And I just want to point out that only around 3% of people had serologic evidence of prior infection at the time they entered into this study, and so there weren't enough people and there weren't enough cases to reach any conclusion about just among people who were seropositive, do they get any additional protection from the vaccine? So that's an open question, but certainly if they analyze the data, taking everybody who was enrolled, regardless of whether they were seropositive or not seropositive at the time they were enrolled in the study, the vaccine effectiveness is no different. An endpoint of interest was the vaccine effectiveness against, quote, severe COVID, and that was based on FDA criteria, based on oxygen saturation and mechanical ventilation and some other criteria. It did not require hospitalization, however, and the overall vaccine effectiveness was 66% against severe COVID, and again with short-term follow-up and the relatively small number of severe events, they didn't have significance on that pre-specified outcome. However, they did do a post hoc efficacy analysis against COVID-19 hospitalization that was on request by the ACIP, because that was hospitalization was an outcome that ACIP wanted to see. And when they looked at hospitalization or admission to the ICU, intubation or mechanical ventilation or death, and you look at the top here, the vaccine efficacy was 100%, but it's only based on 5 cases in the placebo group and 0 cases in the vaccine group. So again, extremely wide confidence intervals, but certainly in the direction we would expect or hope to see. And on the bottom, it shows the first severe occurrence based on of hospitalization anytime after dose 1. So it didn't require completion of the 2-dose series. And if you look at that, we have more events. We had 14 in the placebo group and 1 in the vaccine group. And so that was a vaccine efficacy of 92.9% with a significant confidence interval. Okay, now I want to switch gears and talk about safety for a few minutes. Safety is obviously extremely important, and the FDA had specified the criteria they needed for safety, and the FDA wanted to see a median of 2 months of follow-up after the second dose for safety concerns. And in this study, they had over 37,000 people with a median of 2 months follow-up, and they had actually 19,000 with at least 2 months of safety follow-up. And they also had 8,000 that were what they call the reactogenicity subset, and those are people who completed a daily symptom diary for a week after they received the vaccine to assess local and systemic symptoms. So local and systemic side effects are very common with this vaccine. Pain, local pain at the injection site, fatigue, headache, muscle aches were all very common. Chills were common. Fever was present in around 15%. In general, these symptoms were a little bit less common in people over 65, and they were less common after, or excuse me, they were more common after dose 2 and less common after the first dose. Most of these reactions are mild to moderate in severity, and they resolved in 1 to 2 days. About 8% of people had a grade 3 reaction, which means it was severe enough to affect their normal daily activities. So in general, this vaccine looks to be, I would say, more or less similar to the shingles vaccine, Shingrix, which has been available for a few years now and has an adjuvant in it. And patients are warned that this is people develop frequent side effects to the vaccine. Be prepared for those. They will go away. They're not serious, but it's important that people understand this. In terms of serious adverse events, there were no signals at all. And this table just shows the overall risk of any serious adverse event, and it was essentially the same with no meaningful difference between the 2 groups. And when they looked at particular categories of adverse events, there was no suggestion of any imbalance that was meaningful. I also would note that there were no serious allergic reactions noted immediately after vaccination in the phase 3 trial, although we'll talk in a few minutes about what's happened since then. But I would also note that people with a history of severe reaction to a vaccine were excluded from the trial. Okay, now I want to switch gears, and I'm going to run through Moderna. And a lot of this, most of this was presented yesterday, and it was first posted on the FDA website on Tuesday of this week. So this is all very new information. The Moderna vaccine is shipped and stored at -20, so it's an easier vaccine to handle and administer, and vaccine clinics can deal with this much easier than the Pfizer vaccine. It is refrigerator stable for 30 days. It's 100 microgram IM administration, 2 doses. In this case, it's 0 and 28 days, and the phase 3 trials included people 18 and up as compared to 16 and up. So I'm not going to go through the preclinical development on the Moderna vaccine. I did show you that there have actually been a number of other Moderna vaccines in development with phase 1 trials on other Moderna vaccines. So Moderna's been working in this arena for a number of years. This slide is just published recently in the New England Journal of Medicine, and what it shows is antibody titers by time from vaccination on the x-axis, and the arrows show when vaccines are administered, the first dose on day 1, the second dose on day 29, and shows 3 different age groups, 18 to 55 on the left, 56 to 70, and 71 on, over 71 on the right. So you can see, if you just sort of visually look at this, that the response is really very similar in people over 71 compared to people 18 to 55. And it is maintained over time, at least until 119 days after initiating the series. And so for at least that length of time, we seem to have stable antibody titers with no meaningful reduction. And we see similar responses even in the older age groups, which is very encouraging. Now I'm showing an ELISA here, but if we look at neutralizing antibodies, it's pretty much the same story. So now I want to switch and talk about the phase 3 trial enrollment for the Moderna vaccine. They randomized 30,420 individuals 1-to-1 to either receive their mRNA 1273 product or a saline placebo. 25% of their participants were 65 and up, 21% were non-white race, and 17% had a comorbid condition and were under age 65. So they tried to get a diverse group of individuals, including people at high risk of serious COVID complications. So here we have the primary efficacy analysis. The primary endpoint at the top was symptomatic COVID-19 illness among people who were, had not been previously infected. The vaccine efficacy was 94.1%, and the lower confidence interval was 89.3%. You can see that it was 95.6% in people under 65, and it was 86.4% in people 65 and up. When they looked at subgroup analyses in people with and without comorbidities, they just sort of look at the forest plot here. On the right, the vaccine efficacy is shown on the x-axis, so 100 is 100% protection. You can see that we see a high level of protection sort of across the board when we look at it by gender, with comorbidities, by race and ethnicity. We do see some wider confidence intervals shown by the horizontal lines here in older adults. But overall, we see pretty consistently high vaccine efficacy. And this is very similar to the Kaplan-Meier curve I showed you for the Pfizer vaccine, where we see the placebo group increasing beginning, actually, again, around 2 weeks after the first dose. We see divergence, and we see stable and low level in the vaccine group after randomization followed out to more than 100 days. So the other interesting thing is they would like to know whether or not there's protection against infection as well as illness. And the trials were set up to really assess protection against symptomatic illness, but they did have some interesting data because they collected a swab from individuals just before they got their second dose of vaccine. And among people who were asymptomatic, they found that people who got the placebo were substantially more likely to have asymptomatic shedding of SARS-CoV-2 based on PCR just before they got their second dose, which is suggestive that the vaccine may be providing some efficacy for preventing asymptomatic infection. And if that's true, that suggests that it may be effective for reducing transmission, which is obviously something that is a very important outcome in addition to reducing illness. Okay, in terms of local and systemic side effects from the Moderna vaccine, it's very similar to the Pfizer vaccine, where we see very frequent local and systemic effects like myalgia, headache, fatigue, arthralgias, chills. These are mostly mild to moderate, and they resolve after a couple of days. The FDA presented a risk-benefit assessment in the context of the proposed EUA, and this is for the Moderna vaccine in particular. And the FDA concluded that there was a reduced risk of confirmed COVID-19 at least 14 days after completing the 2-dose series in people without prior infection. And the reduced risk of confirmed severe COVID-19 was identified after completing the regimen. and the findings were consistent in subgroup analysis. So those are the benefits. In terms of the risks, it was pretty obvious there's a lot of reactogenicity. There were a few serious adverse events that were deemed possibly related to vaccination. There was one person that had some intractable nausea and vomiting that was temporally associated. And there were 2 people that had facial swelling that was temporally associated. And interestingly, they had both had people had dermal fillers. This was localized facial swelling. It was not anaphylaxis. There were no other signs of any allergic reaction, and the symptoms did resolve. Serious hypersensitivity reactions have not been reported in the study, but as we'll talk about, have been reported for Pfizer vaccine. And they didn't identify any other safety concerns except noting the limited duration of follow-up for risk assessment and the fact that pregnant women were excluded. So this is sort of the high-level summary of both vaccines. And this is my summary. I want to be clear, this is not an FDA conclusion or a CDC conclusion. This is just my take on this. Both vaccines have high short-term efficacy with vaccine efficacy greater than 93%. And this is consistent with the fact that they generate strong neutralizing antibody titers and a strong cellular immune response. And this is a component that a lot of people, I think, don't realize, but it's not just antibodies. It's the cellular immune response also leads to protection. And that, in fact, suggests that the protection is going to last a longer time. Even if antibody levels decline, protection may continue from the cellular response. We see consistent and high protection in older adults and high-risk subgroups. We see evidence for protection against severe disease. We have frequent local and systemic side effects, especially after the second dose. About 10 to 15% may have to reduce their normal activities because the effects are significant. But no serious safety concerns, and both of these vaccines have had transparent and thorough review by an FDA advisory committee. Now, you've probably heard there have been reports of severe anaphylactic reactions in the UK and Alaska after the Pfizer vaccine. As I noted, there were no episodes during the trials in either trial. And CDC is currently recommending that people with a history of severe reaction to vaccines or injectable drugs should view this as a precaution. They still may choose to receive it or not receive it. If they do receive it, they should be monitored for at least 30 minutes afterwards. At this time, there's no special precautions for people with a history of food allergies, latex, environmental allergies. But these recommendations can change as more information becomes available. So this is a dynamic situation. There are a number of unanswered questions that we don't know yet from the trials. We don't know if the vaccines affect shedding and transmission, although I presented some very preliminary data suggesting that they might. We don't have long-term safety and effectiveness data yet. We don't know very much about prevention of hospitalization and death, although the data we have seen so far suggests that these vaccines are going to be effective to prevent those outcomes. We don't know anything about safety and efficacy in children, pregnant women, or people who are immunocompromised. And we don't yet have a biomarker for protection, which would allow us to make some inferences about protection just based on looking at immune markers. Now, I just also want to mention the CDC Advisory Committee on Immunization Practices. That's another independent group that makes recommendations for vaccines that are authorized or licensed. The ACIP has formally reviewed and recommended the Pfizer-BioNTech vaccine. The meetings are open to the public. They're transparent. The materials are publicly available. The ACIP has also made interim recommendations for priority groups for receiving the vaccine while the supply is still limited. The ACIP uses a very structured approach to assess the benefits and harms called GRADE. I'm not going to go through that for time. And then they also consider other things such as implementation issues and ethics and equity issues in order to help guide their recommendations. The ACIP is meeting tomorrow to discuss the Moderna vaccine. I expect a vote on that tomorrow in terms of Moderna. And then they're meeting again on Sunday to address the issue of priority groups for phase 1B and 1C. And CDC has also provided clinical guidance, and I encourage you to go to this website on top if you have questions about sort of implementation issues. I just want to highlight a few things. These two vaccines are not interchangeable. Whatever vaccine you start with is what you should finish with. The vaccine should be offered regardless of prior symptomatic or asymptomatic SARS-CoV-2 infection. In particular, there is no recommendation to get serologic testing. That doesn't add any value to your decision making. You should get the vaccine regardless. These vaccines should not be co-administered with any other vaccines. We have no data on the safety or efficacy in pregnant women or people with immunodeficiency. So that's a personal decision that each person should make in consultation with his or her healthcare provider. And it's recommended people who were treated for COVID-19 with monoclonal antibodies or convalescent plasma should defer vaccination for at least 90 days. And persons with known SARS-CoV-2 exposure should not be vaccinated until their quarantine period has ended. All right, so I just want to sort of wrap up with a little bit about what happens next. There are a number of systems in place to monitor vaccine safety in U.S. populations. On the left-hand side of this slide are some passive reporting systems, including the Vaccine Adverse Event Reporting System, or VAERS, and both healthcare providers as well as the public and manufacturers are asked to report suspected cases of adverse events into this system. CDC has a system called vSafe that will actively enroll healthcare workers during the early phases and follow them for any side effects or problems. And then there are some other systems as well. On the right-hand side, there's a whole variety of federal systems in place. They're going to be doing monitoring in near real time using large linked databases to look for signals for vaccine safety issues. And I want to in particular mention the Vaccine Safety Data Link, because Marshfield Clinic Research Institute is part of that. We've been part of that for 20 years, funded by CDC. 9 sites link their immunization and outpatient and inpatient records to look at vaccine safety issues. We have data on over 12 million people per year. And Marshfield is actually going to be leading or co-leading 2 studies related to COVID-19 vaccine. One of them deals with a weekly analysis to determine if certain pre-specified adverse events are occurring more often than expected in people who receive the vaccine. And then a second study to look at any evidence of vaccine-mediated enhanced disease, or VMED, and also to look at vaccine effectiveness for hospitalization. I'm not going to say any more about that just because of time. And then lastly, I think it's really important to pay attention and be concerned about vaccine hesitancy and public trust. A recent survey found that 27% of U.S. adults are hesitant to receive the COVID-19 vaccine. This is really not surprising at all, frankly, given how politicized the whole pandemic response has been over the past 8 months. And it's understandable that people have questions. And a lot of this information that's coming out, people haven't heard before and aren't familiar with. And so I think we, as leaders in healthcare and research, have a real role to educate ourselves and educate the community to listen to concerns, to take them seriously, and point people to factual and valid sources of information. And one of them is CDC. This is a poster on the right that CDC has available that encourages people to get good information and to build confidence in vaccination. I just want to mention a few myths and facts that you may hear, as you're trying to give people accurate information. Maybe you've heard some of these. One is the vaccine contains a microchip for tracking. That's just not true. Another one is the vaccine will alter your DNA. It's very well understood biochemistry and cellular biology that these mRNA vaccines do not get into the nucleus and cannot be reverse transcribed to change your DNA. Another one is the vaccine causes infertility. This has been debunked, and there's a New York Times article about this. There's no evidence for this, and there's no biological basis for an effect on fertility. Another myth is that natural infection is better than a vaccine. Well, I think it should be fairly clear that natural infection has already killed over 300,000 of our fellow Americans, including young adults, and children who are previously healthy. And people who haven't died have suffered severe long-term disability. The vaccination is safe, it is highly effective, and it can stop that. And lastly, the vaccine can give you COVID-19. Hopefully I've made it clear why the vaccine cannot give you COVID-19, and the mRNA only codes for a single protein. There are no living viruses used anywhere in the production process of this vaccine. So I just want to end with a quote from my friend and colleague Karen at Hopkins in congressional testimony. She said, building confidence in COVID-19 vaccines is critical because the best vaccine in the world won't work if it isn't used. It begins by soliciting public engagement and developing public trust and open, transparent, independent expert review. So I think what we can all do is, get vaccinated, educate people. The communities will be looking to us to see what we're doing. And if we have a high vaccine uptake, that will be a sign that we trust the science on this. And I can tell you that when it's my turn to get vaccinated, I will be the first to roll up my sleeve and get the vaccine. And I have a son in medical school, and I have a daughter who's a speech therapist, and I strongly encourage them to get vaccinated as soon as they're eligible as well. And that's what they're planning to do. So I think that's what we can do to do our part to protect our patients and our communities. And with that, I want to thank our flu COVID research team. I'm privileged to work with a truly outstanding group of people, and I love it. And I'm so impressed with the work that they're all doing in research and the work that all of you are doing on the clinical side. Thank you very much. Take questions.